genomic pathogen surveillance and some of the challenges that we come up against from a data science and computer science perspective. Cool. So genomic pathogen surveillance is an area of work that really aims to improve the way that we tackle infectious disease outbreaks in the future. In terms of different human health problems that you can tackle with computer science or data science, I think infectious disease is quite an interesting one because unlike a lot of other human diseases, infectious diseases are always changing and adapting over time. Including in response to our attempts to control them. In addition to sort of dangerous new variants springing up and spreading around the world, we're also seeing that whatever treatments we develop to try to treat infectious diseases, there's also quite a pressure on these diseases to develop resistance to them, which creates a constant kind of competition between us and these infectious agents to try and survive and eliminate. I think certainly at the moment with COVID, we think of infectious diseases as affecting us mainly through outbreaks, but there are a lot of other ways in which infectious diseases impact our daily lives and how we can live and survive and thrive in society. Before the invention of antibiotics, things like childbirth, invasive surgery or chemotherapy were extremely dangerous and a lot of people died from infections. So a lot of the way that we practice modern medicine today relies on our ability to identify and treat infections. Also, infectious diseases have a tremendous ability to move and spread internationally quite quickly. So if we don't recognize them early on, they can have a global impact and they can behave quite unpredictably as well. So we don't necessarily know when the next dangerous strain will come up, what kind of changes to its infectiousness or its the severity of the disease it causes might be around the corner. So obviously we want to develop solutions to improve our ability to tackle infectious diseases in the future. There are a lot of ways that we're working on doing this. There's certainly a move to more digitized data capture and collation to try and improve our ability to share information and analyze it effectively. We're trying to change recommendations around the treatments that we use to treat infections so that we can use them more responsibly and more sustainably. We're also trying to improve communication between hospitals and countries. So if something concerning is going on in your neighbourhood, neighbouring hospitals, neighbouring countries might know that there's something they should be looking out for as well. And importantly, from my perspective, there's been a big growth in the use of genomics and genomic pathogen surveillance around the world to get a better understanding of what's going on with infectious diseases. So genomic pathogen surveillance essentially involves sequencing the genomes or the DNA of infectious diseases that might be infecting people, animals, or plants. By sequencing the DNA of these microbes, it helps us to understand how infections that are affecting different people might be related to each other, how the pathogen might be transmitting or spreading from person to person, whether there are intermediates like animals or the environment that we should be taking into account, and also how the pathogen is evolving and changing over time. Is it becoming more resistant to treatment? Is it starting to cause a more severe form of disease? So this is information that we can get from genomic data. We've seen some really exciting advances in how we use genome sequencing in response to disease outbreaks as we've developed different types of technologies. So some of the really cool work that's gone on here in Birmingham in the past was using sort of portable genomic sequencing to respond to some of the really significant viral outbreaks that have happened over the last decade, not just COVID. But for example, in the West African Ebola outbreak, which seems like forever ago now, people at Birmingham were able to take these tiny little DNA sequences, pack them into some luggage along with some other lab gear that they might need, fly to West Africa, set up these kind of prefab labs on site and on demand, and start sequencing the genomes of these Ebola viruses that were coming in from different patient samples. And what that helped us do was not just understand where cases were of Ebola, but also how they were related to each other. And that allowed them to start to assemble transmission chains to see who infected who, and what kinds of long distance travel patterns were showing up as well. So if someone in a remote region had developed an Ebola infection, where did it come from? How did it get to that region? And they were able to start to understand things like how long someone is infectious for, and what the drivers of these long distance transmissions were. For example, they were able to show that when messages got out in the media that made people panic about Ebola, sometimes it triggered them to travel a really long distance to check on a family member. And unfortunately, in that process, sometimes they took the virus with them. So it helped them understand how we might want to change the way we communicate with the public in the future, but also the kinds of infection control measures that are going to be really important moving forward. So it's not just surprise, dramatic viral outbreaks that we're using genome sequencing to monitor. We're also using it for some microbes that we're a bit more familiar with. So this is a collection looking at Klebsiella pneumoniae, which is bacteria that can live on our skin and in our guts quite harmlessly, but it can also cause really dangerous hospital-acquired infections. Particularly if you're undergoing surgery or something like that, you could get a life-threatening wound infection from one of these bacteria. So with the genome sequencing, we're able to look at cases of these infections from all across Europe, see how they're related to each other, and whether there are any strains that appear to have sort of appeared suddenly and then spread rapidly. So they're really common and they're really widely disseminated across Europe. We're also able to look through the DNA and see if there are mechanisms of resistance to antibiotics that we should be worried about. So we can start to see, possibly unsurprisingly, there's a growing trend of antibiotic resistance in these bacteria, particularly because they like to live around hospitals. And over time, we're seeing they're acquiring more and more mechanisms of resistance, sharing them along the family tree to other strains. And it's starting to create a bit of a strain in terms of what treatment options might be available for treating these infections in the future. So my work in this area is really focused on how we can analyze this data better and get more value out of it. So some of my major projects at the moment are looking at using machine learning to comb through that DNA and figure out how we tell a high-risk strain of a pathogen from something that's maybe a bit lower risk. And also recently thinking about really large-scale screening of DNA to try to identify potential hazards, both ones that we are familiar with, that we know we should be looking for, but also ones that might be novel. So this will be a really light touch overview of the kind of work that I'm doing. If anything sounds interesting, get in touch with me and we can talk about it a bit more. So the first topic I'll be talking about is machine learning for detecting high-risk strains of a pathogen. This type of work has really been enabled and really taken off recently due to the fact that our ability to produce lots of DNA sequence data is just constantly improving. The cost of sequencing DNA is dropping exponentially over time. And as a result, the size of databases containing genomic data is increasing exponentially. What this means is that data sets are becoming big enough for us to start using machine learning to comb through them and find interesting. This is kind of reflected in the research activities that are going on in the space at the moment. In particular, in the last few years, there's been a huge surge of interest in using machine learning to analyze genomic data. So you can see both an uptick in papers and citations, really sort of hitting an inflection point around 2018 and 2019. So it's a very popular area to be working in all of a sudden, but because it is such a new area, there's very little consensus on what best practices should be in terms of training machine learning models, testing them and reporting on them in this particular application area. And also, if you look at the citations of these papers to date, most papers that are talking about this research are other papers that are doing this research. So it's a new machine learning algorithm citing an old one or a series of other old ones, rather than scientific work that's taken off as a result of this work or clinical trials looking at the utility of these algorithms. So far there hasn't been a lot of uptake of these algorithms and application in sort of more practical areas. We're all just kind of training machine learning algorithms for the sake of training machine learning algorithms. So I'll talk a little bit about the process that we go through when we try to train these algorithms. The first thing that's really important to think about is how you take genomic data and put it in a useful form for an algorithm. So this is an example of some of the raw data that you get to work with. It's just very, very, very long strings of different combinations of 4 letters, and that's it. There's very little structure, there's very little useful information. This is, I can't remember which pathogen it is, but this is one of my favorite pathogens. If I look at this, I have absolutely no idea what bacteria that's from, what genes are contained in there, whether this is dangerous or not. And I'm an expert who's been working on this for a really long time. So this is really tricky data to work with because it just doesn't look very interesting. Where we start to get interesting signal that we can use for a machine learning algorithm is when we compare the DNA of one microbe to another. And if they're relatively closely related, there are parts that you can line up and identify differences. Usually in one of these studies, you might have a few thousand bacterial samples that you're looking at, and each one might have a few million letters to comb through. And the way that we try to represent the differences between these letters is really important in terms of the kind of insight that you can draw out. So there's basically 2 major approaches you can use. You can pick arbitrarily one of these sequences to use as a reference, and then try to line up all the other samples against it and pull differences. Or something that's becoming a lot more popular is to use a k-based approach, where basically you take your sequence, you shred it into all possible words of a given length, and then you compare those. This is a much more flexible approach. It gets around some of the limitations of saying, if you use the top sequence there as a reference, anything that's not found in that reference or closely comparable to it just gets thrown away. You never see it. Whereas if you look at all the k-mers, it doesn't matter what reference you choose, you're capturing all the information.
Can I ask a question?
Yeah.
So any pathogen can have a base which is only one of 4 characters. Is there any noise associated with each of those bases, or is it highly certain that base is that base?
There are errors in sequencing for sure. So when you sequence a genome, you'll get lots of fragments out, and you know that some of the letters in the fragment are wrong. If you produce enough fragments, it's fairly random where the mistakes are, so you can start to stack them on top of each other from the same sample and figure out what the consensus is. But it depends a little bit on the budget of the project, the quality of the lab work. Sometimes there'll be chunks missing, or a bit where you've only got one read covering it, so you don't know what's right and what's not.
So this idea of just differencing itself may not be good because it could be just measurement noise or something else. Okay, thank you.
I would say the dominant approach today in analysing this data is this k-based approach, so that's what I'll be talking about. K-mers are a really flexible representation of DNA diversity. But if you use this approach, you might wind up generating a really large data set. So you might have 10 to 100 million columns and 4,000 samples, for instance, which statistically is not a great position to be in if you want to separate signal from noise. Something that's happened a little bit more recently is, I think, quite a nice, clever way of reducing that data set without losing any signal. Basically, for a given word length, if you've taken all your sample DNA, broken it up into words of a certain length, those words will overlap sometimes where they're part of a longer fragment. So if you look across your data set and you see 2 words that overlap and they're present and absent in all the same samples, you can consider that information to be redundant, and you can collapse those k-mers into a longer fragment. So if you go through your whole data set and you do this, you can get quite a nice reduction in data set size without any loss of information. So I've got some examples here of some of the data sets that I worked with, where you might cut down from 92 million columns to 4.5 million columns, which is quite a nice reduction. It's still not great, but it's progress at least. What's really nice about this approach, though, is it takes your words and it starts to connect them together in a graph of where those words appear in genomes. And that can give us some really interesting information to work with. So it's collapsing of k-mers into longer fragments. We've decided to call the longer fragments unitigs. And this unitig type encoding, it's just cropped up in the last couple of years. It seems to be a really interesting data structure to work with when trying to understand pathogen genomes. I hope you can see some of the structures on the right there, at least you can see the top ones. So basically, in your data set, you will have a massive graph where each node is a unitig or a word. And those nodes will have different presence-absence patterns across your data set, but they'll be in some interesting kind of a structure. Once you've got all these nodes, you can then test each node for a statistical association with a trait that you're interested in, like drug resistance. When we do this, we can see some interesting patterns appearing in the graph, like there'll be regions of the graph that are positively associated with the trait, or there might be different bubbles in the graph or different variants of the sequence, some of which are negatively associated and some of which are positively associated. And when we look at how those nodes connect together, we can start to see longer sequences of a gene or a regulator that gives us some sort of theory about why those associations might be happening. But unfortunately, to date, while we've got this beautiful graph structure, which is very interesting, every node in the graph is treated as independent, and the whole thing is turned into a table where we've just got one row per sample, one column per node, and then a column for our trait. So no information about the structure of the graph is captured in the data that goes into the machine learning algorithm. It's just a table.
Sorry, can I ask? So I know what the nodes are. What are the arcs in the graphs?
The edges.
Yeah. When do you draw an edge between two unique things?
It's when. Actually, that's not a good example. The edges are where there's some degree of overlap between the words.
So it's structural, it's not functional. So the connections between the nodes.
The overlaps in the words are going to happen in parts of the genome where in some isolates you have genetic differences. So the edges might sort of connect together a gene or a phage or a regulator or something like that. So neighborhoods in the graph will represent a larger functional component.
Right. Okay.
Okay, so once we've got our table of data, we can then start thinking about training and testing the machine learning algorithm. So it'd be good to reflect on what we would want a machine learning algorithm to do if we were to train one. From my point of view, the things that I'm interested in is having an algorithm that can accurately predict a trait of interest from the DNA of the infectious disease. I would like it to do this by capturing the differences in the DNA that actually cause the trait. And I would like the algorithm to be able to generalize to sort of samples from patients that may be from different countries, different hospitals, different patient populations. So whatever it's learning to predict the trait needs to be just as useful in a hospital in the UK as it is in a community setting in Sub-Saharan Africa, for example. And so from a more practical perspective, a good algorithm should help us make better clinical decisions. For instance, what antibiotic to give a specific patient or who to pay special attention to because they're carrying a particularly dangerous strain, so they might need extra isolation in the clinical setting. So that's what I would like to happen. In practice at the moment, I don't think we are there yet. In particular, what we're starting to see now that there's been a big uptick in algorithms being published is that where the algorithm published is published in the way that you can actually run new data through it, which is not standard, but it does happen sometimes. If you apply that algorithm to your own data, you might find that it's not anywhere near as accurate as it appeared to be in the literature. So this is an example of an algorithm that came out relatively early and generated a lot of excitement. It takes that Klebsiella pneumoniae, dangerous hospital infectious disease. You run the DNA of an isolate through this, and it said it could predict with 92% accuracy which antibiotics from a panel of 20 clinically important antibiotics a strain would be resistant to, which could mean that you can correctly treat an infection the first time, and you won't run into treatment failures because you've given the wrong antibiotic. So that's quite exciting. So this algorithm was trained with Klebsiella pneumoniae collected from hospitals in America. And I took it and I tested it on some of our isolates that we had that were collected from hospitals across Europe, and looked at the accuracy of the predictions. So in the table, the numbers on the left tell you how accurate the algorithm was in the paper when they trained and tested it on American isolates. So it was all kind of, the worst performance was 78% accuracy for one drug, and then the best is 97% accurate for another. But when I tested it on our European isolates, the accuracy ranged from 18% to the best was 92%. So in some cases, the accuracy was much, much lower than they said it should be. I've heard other people saying similar things, that they downloaded an algorithm, they were going to use it in their own study, but then it was so terrible that they had to abandon the project because it just wasn't producing anything useful. So why might this be happening? There are a few reasons why these algorithms might perform poorly on new data. One is that the new samples from Europe might just be really genetically different to the samples from America, and therefore it's difficult to generalize. Another reason might be that the algorithm is predicting which training samples your isolate is similar to, rather than whether your isolate carries a genetic difference that would cause resistance. Or maybe the algorithm is overfit to noise in the data, experimental error, bias, things like that. So there are a few possibilities. But one that I'm particularly interested in is the patterns of relatedness between different bacterial samples that you might see in a clinic. So any pathogen that you might come across, all of the isolates of that pathogen present in the world have all come from one ancestor at one point in the past. With bacteria, the way that they reproduce is that you have a microbe, it copies its DNA almost perfectly, and then it divides into two. So over successive generations, you start to get these samples building up that are almost identical, except for a minor difference. And across successive generations, this gives you some nice data in terms of there will be a small number of genetic differences. One of those might match to a difference in a trait, which is great for machine learning, but it also creates some statistical challenges because your samples are not independent of each other. And a lot of your mutations are going to be highly correlated with each other because of this replication process. So it might mean that there's a causal mutation in your data that you're looking to identify, but there's also another non-causal mutation that has almost an identical profile across your data set. And it's really difficult to get away from that problem. So the fact that all of the columns in your data set, regardless of how you encode them, they're going to be highly correlated. This is a problem that we've known about for a really long time. In 1985, when people were just starting to think about mathematical approaches for comparing DNA and figuring out what's related to what, it was pointed out that any infectious disease genomes can't be treated as drawn independently from the same distribution. So samples in your data set are not independent, which from a statistical standpoint creates some challenges and means that a lot of the statistical tests that we would like to apply to this data aren't really appropriate. So this is kind of a cartoon drawing of how your data sort of evolves and changes over time. You have like one data point that turns into two data points that are nearly identical, which turns into another two and another two. This is what that data actually looks like in practice in a real population of bacteria. So if you take all the genomes of your samples, you can hierarchically cluster them according to their similarity overall. And at the root of this hierarchical clustering, you can imagine there was some kind of ancestor. And then as you go down this family tree, you see accumulating mutations that are common to every member of the group that springs out of that part of the tree. So what we find in a lot of these data sets is that if you collect isolates from hospitals in this case, you'll often find parts of your data set contain isolates that are almost identical to each other. And yet they can be like quite a big grouping in the data set because they might be a really successful pathogen clone. They might be really transmissible, they might be really antibiotic resistant. We also find that traits that we're really interested in tend to cluster within this hierarchical clustering. So here with our clinical isolates, all of these rings around our tree represent resistance to different antibiotics. So the different colors indicate different drug classes. And you can see where we've got these flat parts of the tree, we tend to have dark colors across all of our concentric rings. So these are highly drug-resistant strains that tend to be very successful in spreading within hospitals. What's particularly annoying is that resistance to different antibiotics tends to cluster in the same place because those isolates are circulating in hospital environments. There's a lot of pressure to develop resistance not just to one drug, but to all the drugs, and so that's what they do. This creates a challenge in terms of training a good machine learning algorithm because it means that a really easy way to predict resistance to any of these antibiotics is to just identify a successful clone by any means necessary, not necessarily by learning resistance mechanisms. Also creates a challenge when you're looking across these drugs because some of them have a really simple resistance mechanism and some of them have a really complex resistance mechanism. So a shortcut to learning the complex solution could be to just predict resistance to a completely different drug where the challenge is easier. And that seems to be some of what's happening in some of these machine learning algorithms. So a bit of an illustration of what I would like the algorithm to do. This is what I think it might be doing some of the time. Here's our cartoon tree again. Imagine we have a distribution of our trait of interest across the tree. We have some k-mers that are associated with our trait of interest. If we ignore the similarity of the samples, then this k-mer and this k-mer are both found in 2 out of 4 of our isolates that have the trait that we're interested in. But in one case, the k-mer and the trait have changed independently, which seems to suggest that they are really genuinely linked. Whereas in the other case, one change has happened, which has resulted in isolates that carry the same trait and the same k-mer. So in one case, we've got 2 independent events coupling these 2 changes. In the other case, we've got 1 event, which might mean that these are linked, or it might be any other k-mer in our data set. So what I would like these algorithms to do is to learn that k-mer 1 is important, but to not put too much stock in the idea that k-mer 2 is important. Unfortunately, we don't really have a good way of telling the algorithm to do that at the moment. Another thing I thought I should just raise, I'm not going to make a big deal out of it, but it is potentially problematic, is that when we think about deploying these algorithms in healthcare settings around the world, we're kind of hindered by the fact that implementation of genome sequencing is really uneven around the world at the moment. For example, with COVID, almost half of the genomes that are available publicly were generated here in the UK. The next runner-up is the United States. And then whatever's going on with COVID diversity in the rest of the world, we have a really limited snapshot because there's not a lot of genome sequencing going on, which means that if we want to understand differences in the DNA of a pathogen and how they relate to differences in the trait, we're getting a really biased picture by mostly looking at the UK and the US. We also have a big bias in terms of the pathogens that we're interested in and the amount of DNA that we collect from them, meaning that we've got a great potential to train algorithms for some applications, but we're pretty limited in what we can do with others. Okay, so I mentioned a great way to predict resistance to one antibiotic might be to learn to predict resistance to another antibiotic. There is evidence to suggest that this is going on. So in this paper, some researchers trained an algorithm to predict resistance to a panel of antibiotics, and then they looked at the top predictors that the algorithm was using to predict resistance to each drug, and they partitioned them out into mechanisms that we know are real and are actually associated with that drug versus mechanisms that we know do not cause resistance to that drug, they cause resistance to a different one. And they found that a lot of the time the algorithm was learning more wrong predictors than right ones because all of these traits are correlated and it's really hard to tease them apart. Where this becomes a problem is when you're trying to use these algorithms to choose the right treatment from a panel of drugs. If you're actually predicting resistance to a completely different drug, the recommendations coming out of these algorithms might be kind of useless. And so we need to be learning better mechanisms for predicting for one drug and not for others. Okay. So we've got sort of limited examples we can draw on when we look at real data. So I tried looking at some simulated data to get a better picture of what the strengths of these algorithms are and what the weaknesses are. So for this here, I took real DNA from a population of bacteria, but then I chose parts of that DNA to be causal for a hypothetical trait. So I said this KMA has an effect size of 2 on our hypothetical trait. I did that for a bunch of different parts of the genome, and then I simulated that trait. Then I asked a machine learning algorithm to take that KMA data and predict a hypothetical trait. So I found that if you do that, a machine learning algorithm can do a pretty good job of accurately predicting the trait. But then when you look at the effect sizes it's learned for each of the different KMAs that I know are causal, the effect size and the direction of the effect were just all over the place. So it learned in some cases a really accurate, it learned to make really accurate predictions, but didn't learn anything about the actual biology of the trait or what was causing it. Meaning that even if you've got a really accurate looking algorithm, you shouldn't really try to learn anything about biology from what the algorithm has learned. Okay. How are we doing for time?
10 minutes.
Okay. I might skip this. I wanted to say about this, I raised it early on that when we encode our data, we turn it into a graph, which I think is very cool. What I would like to do in this area is actually to make better use of this graph structure moving forward. What we know about the biology of these traits is that around the genome, there are big regions of the genome that have nothing to do with the trait. They're not causal, not having any interesting interaction with it at all. There are other regions of the genome where there might be a lot of different variants that influence the trait and interact in interesting ways. So I think it'd be very cool when we're learning to actually hone in on different parts of this graph and really pay attention to them, and then eliminate other parts of the graph because they're not very interesting. I haven't completely figured out how to do this, but I know there are other areas of research that work on graphs or data that have some sort of a structure to them. I thought a relatively simple solution would be to train different sort of sub-algorithms on a small region of the graph to see if that region can be used to produce accurate predictions. And anywhere that that region of the graph is not useful, just throw that data away and only keep the sub-models that are actually contributing to some sort of predicted accuracy. Once you've got your sub-models, you could then combine them together to produce sort of a more accurate ensemble. If we were looking at a much smaller amount of the data set, what we could also do is look at which of those KMAs are associated with a successful clone or a highly similar sub-region of the data set, and penalize learning any of these nodes that just predict it's part of this family or it's part of one of those really successful strains. So I think there's a lot of work that we could do in this space. Currently, no one's doing any work with graphs. So any progress on this front would be, I think, quite exciting. I'll see how much of this I can tackle in 10 minutes. So another project that's just come up, which I think is very exciting, but also potentially a huge challenge for us, is screening for dangerous DNA on a much greater scale. So one of the things that became really clear with the COVID pandemic is that early warning of a dangerous pathogen spreading would have been very useful. In hindsight, we realized that some of the first COVID cases probably happened around October or November 2019, but alarm wasn't really raised until December. If we had put in place infection control measures months earlier, we might not be in the situation that we're in today. We've also seen around the world that places that responded very quickly with infection control measures against COVID fared a lot better than countries that sort of delayed. At the moment, a lot of our surveillance systems for detecting outbreaks of infectious diseases rely on looking for something that we already know about, like a flu outbreak or a cholera outbreak or something like that. We're not very good at detecting something that's completely new because we don't know what we're looking for. This is particularly worrying because the majority of viruses that are out there in the world have never been sequenced before. We've only sequenced a tiny fraction. So if we were going to do some sort of genomic surveillance for the next pandemic, we wouldn't really have any idea what we were looking for. So one solution to this problem that's being proposed is something called the Nucleic Acid Observatory. This is essentially just a global network of sequencing machines that are just looking at all of the DNA that's present in, say, wastewater or rivers or communities, just constantly churning through DNA, not necessarily of pathogens, but just of everything, to see what's out there. There's some evidence that this could be a really good approach for pathogen surveillance. We've seen in a lot of cases that DNA from pathogens shows up in wastewater before clinical cases are reported. This has happened with COVID. We've also seen it recently with an outbreak of polio, where they detected polio in the wastewater. They rolled out a massive vaccination campaign in a potentially vulnerable population, and polio did not take off, which is fantastic. It means that it was an effective early warning measure. So the idea is, what if we did this all over the world, all the time, just monitoring DNA to see what's coming up? So this would involve metagenomic sequencing, which is basically where you take a sample. It's not of a culture of a microbe from a person. It's not pure. It doesn't contain just one thing. It contains heaps of things, particularly if it's wastewater. It'll have plants, it'll have us, it'll have a whole bunch of microbes that we're not necessarily interested in. And we'll just be sort of sampling some of the DNA of that and sequencing it. So whatever we do and however much money we spend on this, we're probably going to generate a lot of sequences, but only really a small fraction of what's actually in the sample. On top of that, we know that whatever method we choose to produce this DNA might introduce some sort of bias or some sort of misleading signal. We know that this type of data is really hard to process. I showed you a genome from a bacterium before. This is what you might get from a metagenomics experiment, where each color, I've imagined, is a different organism. So sometimes you get multiple fragments from the same organism. Sometimes you might just get one. We've also got some additional information here about how confident we should be in each of these letters, how high quality each bit of this DNA sequencing is. So this is the raw data that you have to process at massive scale in real time all over the world. So a lot of challenges in this type of work. One of them is cost. This would be extraordinarily expensive, but another one is what you do with that data as it's coming through. How do you find something important? It's an enormous amount of DNA. There's a spatiotemporal component that you have to take into account. And there's also, you know that the data you're looking at is extremely incomplete. Another issue is how do you store and share the data? Keeping all the raw data would be really expensive. This would be like terabytes and terabytes and terabytes of data constantly streaming in. Sharing between sites would also be really difficult and potentially very expensive, particularly if everyone's sharing with everyone. But there could be some serious benefits. You could detect new threats as they appear rather than as they sweep through hospitals and cause lots of cases. You could also look backwards in time if you realize an outbreak is going on and say, how long has it been going on for? Where is it in the world? Which helps you understand how it's spread and what you should do next. And you can also flag when you should introduce a control measure into a new location because that infectious disease has just arrived there. But say you were looking for the next COVID, the next thing that you don't necessarily know that you're looking for, how do you know when you found it? How do you flag something concerning? The idea that's been put forward is to look for an exponentially increasing k-number. So just a shred of sequence that wasn't around much before, and then is suddenly showing some kind of exponentially increasing pattern. If you did that and you had some sort of event flagger that said something is increasing exponentially, you would then have to figure out what it is and what you should do about it, and if it's even a problem. So this is the idea that I'm thinking about at the moment in terms of like, is this a good idea? Is it feasible? How would you process this data? One idea that I had that might make this a little bit easier, more robust, more useful, is to apply that same graph-based approach. So some researchers have actually made a graph, not just of a species of bacteria, but like all the bacteria ever. So there's a mega graph. If you then took the k-mers from your sampling and you mapped it to that mega graph, because you're only ever looking at a small subsampling of the DNA in a sample, the idea that the exact same k-mer is going to show up in all your samples and show that exponentially increasing pattern might not be very robust. But if there's a part of your graph that represents a novel virus, you might find that in different sites in space and time, that graph, that region of the graph starts to show some interesting activity. So this is something that I'm thinking about at the moment in terms of what's the footprint of this in terms of storage and sharing, how quickly could you map new samples to this graph, and how robust is this method for detecting exponential spread of something. So it's kind of the next thing on the agenda that I'm looking at. Some of the questions I have around this is, say we do identify an exponentially increasing k-mer. Is that a common phenomenon? Does it mean it's an infectious disease? What other pieces of DNA might show this activity? And should we take it seriously if it occurs in real life? And also, if we do see something exponentially increasing, how well can we extrapolate from that short sequence what we're looking at? Is it a virus? Is it a bacteria? Is it sort of pieces of DNA that we know are very good at copying themselves and jumping around but don't actually cause human disease? So there's a lot of scoping out to do here. It's still a very new idea. I think it's gaining a lot of support in terms of what we might implement in the future to tackle the next pandemic. But there are a lot of sort of data analysis and processing questions that need to be answered to see if this is actually worth investing hundreds of billions of dollars into. So yeah, hopefully I've mostly kept time. Thanks for listening. I've put my email up here in case you wanted to ask follow-up questions or more. But yeah, also happy to take questions now.
Yeah, so on the last part with the exponential growth, so has anything been done like that before where people just look like at k-mers and see how do they develop over time?
No.
I know like with the COVID, somehow like they do like this wastewater analysis for
Yeah,
example, right?
if it's reference-based,
They look for one particular kind of.
yeah, so the plot I've shown here,
Okay.
this is broken down into sort of very large groups of bacteria. So when you look at all the bacteria in the world, you can kind of group them into some major clusters. This is any reads mapping to anything in that entire family of bacteria, rather than one k-mer. So we know that you can see exponentially increasing patterns when looking across a huge amount of DNA. But does that same signal show up when you're really restricting yourself to just like one read, one short word? We don't know.
And why would it be so expensive to do that?
Metagenomics is just really expensive.
Okay.
And it's the scale as well, because you can imagine if you just sequenced one wastewater point live, that would be kind of costly. But on the scale to do it all over the world in a way that's meaningful, it would be really expensive. So I think the first pilot of this is just going to be carried out in Boston, and that's going to be multiple sites running live all the time. The more DNA you sequence, the better your surveillance system is going to be, but the more expensive it is as well. So I think they've got like a tiered pricing model.
Okay.
Scales with effectiveness, and it's going to be an iterative process of saying, if we did it in a really small place, can we handle the data? Does it make sense? Is it flagging exponentially increasing k-mers all the time and therefore not very useful? Or does this look like a viable model to sort of invest in more?
I have a question. So in terms of this data collection and do these sequences, right? And until you get your data into your computer, how long it will take? Like, do you think if it's slower than this exponential growing, then there's no point to do that, right?
No, it depends a little bit on the machine that you're using and the workflow that you're using. If you're using a MinION sequencer, which is what the researchers were using to look into Ebola, that's kind of live. You put a sample in and it starts producing sequence immediately. And you can sort of run it for as long as you want and you'll get more and more data out of the sample. But you can get meaningful data within half an hour.
Okay.
It's a common more than a question. I didn't understand 10% of that. But it reminds me of some of the work that Leandro is doing. Have you spoken to Leandro Miku? Because I think it's Leandro that did something similar, but with financial market data, whether there's lots of the data and they try to build a model based on what's coming in, or something like that. So you should definitely, I'm not sure if Leandro is online here, but Leandro would be someone that would be really interested in problems.
So can I also ask you about this? So the non-IoT nature of the data that you spoke about is actually quite interesting. So can we assume that we have information about these almost like phylogenetic trees?
So we do our best to infer sort of the family history based on, you look across the whole sequence and you try to figure out what's the most likely scenario that would explain all of the similarities and differences. We are often not highly confident in the whole tree. There will be bits that we're really confident in and bits that we're like, could go either way. So we have some, it's better than nothing.
Right. Because, like in machine learning, there are methods that basically say, if you know roughly what is the dependency between different data items, then you can like take care of it in some sense. So the usual example is time series, where you know that things are more dependent than more recently are, and that has been generalized to graphs, which is like time series is like a trivial graph. And that has been generalized, so we have like heat kernels and other things, basically saying if your point is here, then if you have neighborhood on the graph, they are more dependent. It's exactly what you showed us.
Yeah, I think the time series data is the closest I've been able to find to what we've got here. Because in some cases you can actually go back in time, and it looks a bit more like a time series. Often what you've actually got is something that goes back maybe like 100,000 years, and you've got a lot of things at the same time point.
Yeah, so there are, this has been generalized to graphs.
So I think some of the better analysis methods for producing robust results will sort of go back through that hierarchical clustering and create nodes in the ancestral spots based on our best guess of what they look like. And that gives you much better results than if you just look at all of the tips of the tree. It really, really depends on the quality of the, of the reconstruction though. But I think as we get more samples, hopefully the quality of our reconstructions will get better.
You can like even bootstrap yourself. You can try to learn from your performance about things in the graph that you don't know. But anyway, this is for another discussion.
Something that I didn't bring up actually is something that is really promising, but also potentially a bit concerning, which is that now we have sort of machines you can buy. You say, I want to make 10,000 edits to this genome, and you stick your bacteria in. 2 days later, 10,000 point edits come out. If you could get the machine learning algorithm to give you hypotheses of, I think if you change this, it will be resistant. Pairing that with a machine that can do that would be a really cool way of learning, but also creates the concern of like, if you're trying to predict what would make a really dangerous pathogen, that capability will be available to people very soon.
Try to do some reinforcement learning.
That is something like that I think appeals to a lot of people. It will be possible, but it creates the concern that like, how would you account for the harm that could be done by that sort of process if the wrong person was doing it, or if you had an accident and something that you may have got out of the lab.
Do we have questions from? Okay, so Jim.
I just wonder, how does the length of the chimeras affect what information you get, say in the last method? Because would sampling different length chimeras lead to spikes at different levels that could indicate maybe?
Yes and no. I think when we've looked within one species, chimer length kind of affects the complexity of the data, but not too much. So some people want to look at a chimer length of like 5. Then you have far fewer possible chimers, but the signal that you get out is not very useful because by chance, chimers of length 5 can just appear in completely different things. Normally, when you're looking within a species, a chimer length of like 30 to 40 gives you precision. We can say this is probably from this virus rather than like it's from something alive, probably. And it gives you a nice graph structure that makes sense. But the longer the chimer, the more specific it is to like one strain. The shorter the chimer, the more you can do like species identification.
Okay, yeah. So could that be used, say when you get all your data and it's all mismatched in the...
Is it a hyperparameter that you could choose? One of many.
It's always tuning hyperparameter. Cross validation.
Yeah.